Well, good day to everyone joining us and welcome to today's X Talks webinar. Today's talk is entitled Nitrosamine Impurities in Rifampin. What does it mean for your drug-drug interaction study? My name is Ryan Muse and I'll be your X Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using the same chat panel. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Celerion, who developed the content for this presentation. Celerion, a global leader in early clinical research services, offers a unique combination of medical expertise, clinical operations experience, and scientific excellence that gives their clients the confidence to make fast, accurate decisions about their drug development path. For 50 years, Celerion has leveraged the latest operational concepts and technologies to execute safety, tolerability, pharmacokinetic, and pharmacodynamic studies in highly controlled clinical environments. Now I'd like to introduce our speakers for today's event. Dr. Sabina Palialunga serves as a subject matter expert in metabolic diseases such as NASH and type 2 diabetes. She has published extensively in 3 areas, as well as on topics related to pharmacology and drug development, which also includes drug-drug interaction studies. She joined Celerion in 2015, and prior to that, she was in academia. Sabina received her PhD in biochemistry from McGill University, Canada, and her training also includes 3 postdoctoral fellowships. And Dr. Arnott Van Hurst is a subject matter expert in Celerion's respiratory research team and supports early phase drug development programs across all indications, including clinical pharmacology studies such as drug-drug interaction studies. Arnott joined Celerion in 2015 and has over 25 years of drug development experience of biotechnology companies such as CROs. He received his PhD from the School of Medicine at Leiden University and is qualified as a clinical pharmacologist in the Netherlands. Together, Sabina and Arnott published a mini-review last summer in Clinical Pharmacology and Therapeutics, addressing today's topic of rifampin use in drug-drug interaction studies. Now, without further ado, I'd like to go ahead and hand the presentation over to our first speaker today, Dr. Sabina Palialunga. You may begin when you're ready.
Thank you, Ryan, for that introduction, and good morning, everyone, and good afternoon. I'd like to thank you all for joining us today as we present nitrosamines impurities in rifampin. What does it mean for your drug-drug interaction study? Now, before we begin, I'd like to just first introduce you to Celerion. We are a full-service CRO and predominantly work with innovative biotech companies as well as large pharmaceutical clients. Our strategic focus lies within early clinical development drug phases, and specifically first in human through proof of concept studies, as well as labeling studies. Our bioanalytical laboratories focus on large and small molecule drug development, and we also offer full service data management and biometrics. For multi-site or later phase patient studies, this is managed by our global clinical development team, which can include our own clinics as well as external sites. Now, here's a snapshot of our clinical experience over the past decade, and it's broken down by healthy volunteer studies and our patient studies. You can see we've conducted an extensive number of clinical pharmacology studies over the past few years and have done quite a few DDI studies, almost over 300 studies. And we are a global company with clinical pharmacology units located within the US, in Lincoln, Nebraska, Phoenix, Arizona, as well as in Belfast, Northern Ireland. Now, to get to the topic at hand, in August of 2020, Rifampin was found to contain nitrosamine impurity of 1-methyl-4-nitrosylpyrazine, or MNP for short. And rifampin is a semi-synthetic antibiotic used to treat tuberculosis. This crowd may be more familiar with it as it's used as a CYP3A inducer for multiple dose DDI studies, or OATP inhibitor when given as a single dose. And the impurities found within rifampin have significantly impacted drug-drug interaction studies. We recently published a review article in CPT addressing the current situation from a drug development point of view, and have recommended alternative CYP3A inducers for continued DDI studies. And today we'd like to share some of the highlights from this paper, as well as address how both sponsors and regulators have responded to the current situation. So nitrosamines are a chemical compound class that contain a functional nitrosyl group. And on the right, you can see some examples of common nitrosamines, such as NDMA and NDEA, as well as the MNP, the compound that has been found within rifampin. Now, the ICH considers nitrosamines a cohort of concern, and that's because they are probable or possible human carcinogens. The nitrosamines can undergo metabolism by CYPs for bioactivation to these reactive intermediates that can bind to DNA and form DNA adducts. This can lead to genotoxic effects and increase the risk of cancer. It's important to note, though, that nitrosamines are common and can even be found in low levels in drinking water, in foods like processed foods or cured meats, dairy products, vegetables, and tobacco exposure. The majority of nitrosamines are carcinogenic. However, a small proportion, about 18%, are not. And we don't actually know which bucket MNP falls into. We don't have direct human data, and the animal data is actually quite discrepant. So that being said, MNP is considered potentially carcinogenic. And since it's potentially carcinogenic, an acceptable intake limit has been assigned. And the acceptable intake level is the daily exposure of a compound that approximates a negligible risk of cancer after 70 years of daily exposure. So the FDA actually uses the NMDA TD value of 96 nanograms per day to calculate the acceptable intake level for MNP. And this is a conservative estimate. So to determine this value, you take that 96 nanograms per day TD value divided by a standard typical dose of rifampin, which is about 600 milligrams per day, and that yields 0.16 parts per million. Anything above this level is considered a concern. And nitrosamines in medication have been making headline news since 2018. First with angiotensin receptor blockers containing a number of nitrosamines above the acceptable intake level, followed then by some batches of metformin as well as Zantac. And then in 2020, we had the rifampin situation, and then last year, Chantix, anti-smoking product, was found to contain nitrosamines as well. And the source of nitrosamines in these medications could be due to several reasons, such as it could arise during the manufacturing product. Secondary amines or nitrites within these chemical compounds, as well as potentially in the water or solvents, could create nitrosamines reacting to nitrosylating agents, or even the excipients themselves. In addition, the chemical structure could become nitrosylated, as in the case with Chantix. And then storage and packaging conditions could also lead to increased amounts of nitrosamines. For example, some blister packing contains amines that could become nitrosylated. So all of this impurities in several medications have led to increases in non-compliance in manufacturing. And this is results from the FDA surveillance data. You can see over the past few years, the non-compliance rate has been slowly increasing and even jumped to 35% in 2020. And it's interesting to note that nearly 1/4 of that 35% is due to nitrosamines in various medications. So what are the regulators doing? Well, actually they released some guidance in order to control nitrosamines in both marketed and drugs in development. And the EMA, FDA, as well as other regulatory agencies have taken a harmonized approach by creating a 3-step process. And the first step is a risk assessment, where drug manufacturers are to evaluate the formulation and manufacturing process to identify any risks. And this would be based on any potential amines or nitrosating agents within the manufacturing and drug development process. The next step is to confirm testing if a risk is identified through the development of sensitive and specific assays. Then, if those are again positive, to implement changes in the reformulation or manufacturing to remove or lower the presence of nitrosamines in these drugs. Now, the first step has been completed, and the second and third step should be completed by next year, according to the FDA and EMA guidance. And the EMA has actually published some of their findings from that step 1 process. Here you could see the findings for chemical drugs as well as biologic drugs. And 60% of chemical drugs were found to potentially hold a risk of nitrosamines, whereas 1% of biologic drugs. So the manufacturers identifying risk will then go through the step 2 and step 3 process if needed. And I don't believe we will find more information until the deadline is set next year. But these next steps are definitely underway. Next, we'll discuss the potential root causes for nitrosamines in rifampin and how it's affecting both patients and the drug development sector. As I mentioned, rifampin is a semi-synthetic product derived from A. mediterranea, which is a gram-positive bacteria that naturally produces the antibiotic rifamycin B. Rifamycin B undergoes several reactions, and in the last step, AMP is added to yield rifampin. There's a potential that AMP could be oxidized by free nitrates or by air oxygen to form MNP as its byproduct. And this could result in the MNP levels being over that 0.16 parts per million. Another potential source of MNP in rifampin capsules may be related to thermal degradation. So here you can see over 30 days, MNP actually increases by 25% when stored at 40 degrees Celsius. And that value actually nearly doubles when stored at 60 degrees Celsius. And while temperature is strictly monitored during the manufacturing phase, it's likely not a contributing factor to the current situation, but could become an issue if sustained temperature versions occur during transportation or storage of the medication. Now, despite MMP being above the acceptable intake level of 0.16 parts per million, the FDA said that they would not object to batches as long as the levels were below 5 parts per million to be distributed for patients. And that's because the risk of tuberculosis outweighs a theoretical risk of cancer. Rifampin is actually considered an essential medication by the World Health Organization. And complications to tuberculosis could include lung damage, spinal pain, meningitis, liver or kidney impairment, and even death. In January of this year, the FDA actually published results of current batches, and while no brands were found to contain MMP below the 0.16 parts per million value, you can see that current ranges are below the temporary level of 5 parts per million. Now, another issue compounding this situation is that there's currently a rifampin drug shortage. Several states in the U.S. have issued health advisories about this shortage and outlined guidance to provide and prioritize patients that are at risk. And the short supply is partly due to current medications being past the expiration date, as well as a shortage in the active drug due to supply chain issues. You'll note that there are limited batches available, and one of the pharmaceutical companies have actually discontinued manufacturing of the drug, which is compounding the drug shortage issue. So what can the industry do? Well, immediate efforts would include increasing production to relieve the drug shortage for those manufacturers that are still producing rifampin, and then to test each batch, make sure that the levels are below this 5 parts per million value. For long-term efforts, changes to the manufacturing process itself could potentially include the addition of antioxidants to the formulation, as this could prevent the oxidation of MMP, for example. Or they could even modify or change the microenvironment of the reaction itself, changing it to a more neutral or basic pH. Acidic environments tend to promote nitrosamine formation. So what about DDI studies with healthy volunteers? Well, it seems like all regulatory agencies have taken a prohibited approach. The FDA states that using rifampin in healthy volunteers is not acceptable. The EMA commented that rifampin containing nitrosamines above the acceptable intake level should not be used in healthy volunteer DDI studies. And likewise, the MHRA indicated that rifampin should not be used in drug-drug interaction studies in healthy volunteers. So next, I will pass it to Arnaud. Well, he will present the sponsor's response to the regulatory agencies halting the use of DDI with rifampin and provide some alternative options for continued drug development.
Well, thank you, Sabina. Now, first, let me take you through how different scenarios for drug-drug interaction studies have emerged in the past 2.5 years, also illustrating how we as Celerion have dealt with the rifampin issue recently. After the announcement of nitrosamine impurities in August 2020, the FDA notified several of our clients that using rifampin in healthy subjects is not acceptable. and they suggested to use either phenytoin or carbamazepine instead. And one of the projects that we were about to start was planning actually to evaluate rifampin as a strong inhibitor, strong inducer in part 1, and subsequently rifabutin as a weak inhibitor in the second part of the study. So following the FDA feedback, the client decided to remove the first part of the study and entirely focus on part 2 involving rifabutin. So this was one way to get around it. But for the US in general, it meant for us that there would be 2 options remaining. One is to change perpetrator and find another one. And the second one would be to basically wait for clean rifampin batches to be available again. However, as there still was a preference for using rifampin as the inducer in interaction studies, and given also the relatively short exposure of the drug in healthy volunteers during a drug-drug interaction study, we considered the option of conducting the study in the UK. And in this scenario, we were thinking of including a risk mitigation approach. As already mentioned by Sabina, nitrosamines are commonly found in foods, as shown here on the right, so like in products like processed meat. And studies have estimated that the average daily exposure from food intake is around 3 micrograms per day, and even many times higher is the exposure in smokers. Now, based on 5 parts per million of nitrosamine impurities in rifampin batches, that would yield in a daily dose of 600 milligram rifampin used over 14 days in a typical drug-drug interaction study, would yield an exposure of nitrosamines effectively of 42 microgram. But in principle, this could be compensated by implementing a low nitrosamine diet, thus reducing also the nitrosamine intake by exactly or by a similar amount of 42 micrograms. So following the FDA rejection of rifampin use in healthy volunteers in the US, we introduced this risk mitigation or a risk mitigation approach early last year in a UK setting. Because we have clinics in the US and in the UK, we are flexible in conducting studies in either geography. So in this risk mitigation approach, we obviously included the risk mitigation as per the labeling of rifampin. But in addition, it was argued that rifampin administration was only for a very, very short duration, with nitrosamine exposure within the limits of acceptable for chronic use. And finally, by also implementing a low nitrosamine diet, the total nitrosamine exposure would thus be within the daily life estimates of exposure according to the studies I just referred to. So for the very first study protocol that we submitted and that included a risk mitigation approach, we actually received a ground for non-acceptance notice from the MHRA. However, they didn't object against our approach, but they requested that a risk-benefit discussion would also be included in the protocol. So in a new protocol, we included a discussion on the risk-benefit balance. And shortly after submitting the protocol, we also received approval from the MHRA without any comments. And this drug-drug interaction study was completed without further complications. So while this strategy with the use of rifampin in drug-drug interaction studies and including also a risk mitigation strategy initially worked out, this approach was eventually rejected by the MHRA late last year. And we've been seeking feedback from the MHRA directly And they didn't provide any detailed argumentation, but the bottom line was that rifampin was no longer, could no longer be used or should no longer be used in healthy volunteers. And therefore, we had to look for alternatives and replace phenytoin rifampin by phenytoin in subsequent drug-drug interaction studies as of early this year. And the first studies that we started were actually completed last summer in one of our US clinics. Now, the final design that I want to touch upon here is a single dose use of rifampin, if that would still be acceptable. I touched upon induction by rifampin of cytochrome P450 enzymes, but if a transport protein is suspected to affect a new drug's routing in the body, a DDI study may also be indicated using a transporter protein inhibitor. And if you look at the FDA website that I referred to previously, there are several options shown which also include rifampin. And actually, one of the advantages of rifampin is that it is only requiring a single dose, but also depending on the metabolic pathway of a particular drug and the impact of any transporter protein inhibitors that one would consider, there may be other transporter proteins or even CYP enzymes affected that are also relevant for the new drug's metabolism. And there may be scenarios where rifampin may be better positions as a perpetrator in such DDI studies. However, would single dose rifampin still be acceptable? After the lessons we've learned, maybe with our interactions with the authorities, we feel that this is no longer acceptable. After all, the impurities in rifampin, the nitrosamine impurities, have never been shown to show a linear dose-response relationship. So there still is an option that the use of rifampin, even at a single dose, can be carcinogenic. So we need to select alternatives for rifampin as the perpetrator in drug-drug interaction studies. And actually, earlier this year, ICH has issued draft guidelines for drug-drug interaction studies, stating that the first clinical interaction study should use a strong index inducer, or in the context here of inducers. And it also mentions that moderate inducers should only be used if no strong alternative is available. So examples that that are mentioned here in the guidance are carbamazepine and phenytoin as strong inducers next to rifampin, but also surprisingly efavirenz as a moderate inducer. Now, in principle, one could consider a slightly broader set of drugs that also induce CYP3A and could potentially replace rifampin in such studies, including several that are also referenced on the FDA website. But in practice, application of most of these is limited by unfavorable safety profiles or, for instance, being relatively weak inducers. Amongst all, we feel that phenytoin stands out as the preferred alternative, but also several other candidates like carbamazepine, efavirenz, as just mentioned, and also lumacaftor could be options, which I will briefly discuss hereafter. Now, I also want to highlight before doing so that the actual pathways of inducing CYP enzymes may be slightly different. So in principle, activation of xenobiotic sensing receptors occurs, which include the Preckinase receptor, the PXR, and primarily also the constitutive androstenedione receptor, or CAR. Like rifampin and lumacaftor, for instance, are primarily activating PXR and thereby inducing CYP3A. On the other hand, carbamazepine and phenytoin, according to literature, primarily activate CAR and to a lesser extent also PXR. to establish the induction of the cytochrome P450 enzymes. Now, to bear in mind, a final issue is that there may be differential effects on liver and intestinal CYP3A, and I will get back to this in the case of efavirenz later. So first of all, carbamazepine. This is a anticonvulsant drug that is, as said, a strong inducer of CYP3A. It has, however, a major drawback, which is the risk of serious dermatologic reactions at high starting doses, and as also illustrated on the screenshot on the right. So this is also why the drug has received a black box warning, and it's particularly relevant in populations in people of Chinese descent, which may have a risk of about 10 times higher in developing such adverse effects. Now, the way to get around this, one can use carbamazepine in studies by mitigating the risk of such adverse effects, for instance, by excluding Asians that have a positive allele that is associated with the risk of carbamazepine hypersensitivity reactions. And secondly, is also a dose titration approach that may be helpful in limiting the early reactions of the high starting doses. And currently, actually, there's quite some experience with carbamazepine as the perpetrator in drug-drug interaction studies. And this graph we've pulled together based on data from published drug-drug interaction studies using carbamazepine, and we considered the exposure fold reduction, or the AC fold reduction, for each substrate. And in as far as possible, we also made a comparison to Rifampin data. So from top to bottom, you will see the different substrates that were tested with increasing sensitivity for CYP3A metabolization, and with the clinical index substrate midazolam marked in the blue line. So this graph shows that carbamazepine is a similarly potent inducer of CYP3A as Rifampin, although the overall effect obviously may depend on the substrate under investigation. The next candidate I want to discuss is efavirenz. I already mentioned that it is a moderate inducer, so that is a drawback. Another downside of using this one is that reportedly it may take quite a while to reach the maximum induction of CYP3A, compared to Rifampin only taking 2 weeks. Moreover, there's fairly limited experience with the drug in drug-drug interaction studies. And back to the moderate inducers thing, reminding you of the ICH guideline, if we have good alternatives that are strong inducers, then those would be having the preference. Now, the final thing I want to bring to your attention is there is conflicting evidence towards the induction of CYP3A in the gut when compared to the liver. And this, particularly for efavirenz, raises doubt on how reliable it would be as an inducer of CYP3A, particularly for orally administered drugs. Lumacaftor then is the next option I want to touch upon. And interestingly, this is the latest addition to the list of CYP3A inducers on the FDA website, added late August this year. Lumacaftor maybe has the downside of only being available in combination with ivacaftor, together named Orkambi. However, the effect of ivacaftor ivacaftor is not anticipated to really impact the outcome of any drug-drug interaction study. And the upsides of lumacaftor are that it is also a very strong inducer of CYP3A, as far as we know it currently. And also it has a favorable safety profile. So unfortunately, there's limited experience so far, and particularly the effect on midazolam, being the clinical index substrate, has never been studied yet. But I believe this may be the new kid on the block and a candidate to consider for future studies too. Then phenytoin, also being an anticonvulsant drug like carbamazepine, and also being a strong inducer of CYP3A. And there have been quite a number of studies, drug-drug interaction studies, in which phenytoin has been applied. Phenytoin metabolism is occurring for approximately 90% through enzymes CYP2C9 and 2C19. So using this drug in a clinical trial would require genotyping for those two cytochrome P450 enzymes to exclude poor metabolizers. Otherwise, the drug has the downside that it has a narrow therapeutic window, so dosing should be very done carefully because there are safety concerns in particularly neurological events. And otherwise, it also has a longer half-life than, for instance, carbamazepine, and it may take at least 2 weeks to get to maximal CYP3A induction. On the other hand, mitigation measures that one can take is exclude subjects that have a history of neurological events or seizures, and also exclude women of childbearing potential because of potential prenatal risks. And finally, it's good to mention that if a substrate is suspected to potentially increase the level of phenytoin in the blood, then it is indicated also to closely monitor phenytoin levels during a drug-drug interaction studies. But the recommended phenytoin regimen would be 300 milligrams on a daily basis for at least 2 weeks, and that would be, or has been shown to be effective. Now, like we did for carbamazepine, we combined data from published drug interaction studies using phenytoin, and also again listed the reduction in exposure of various substrates, and in as far as possible made a comparison to Rifampin data available, shown in blue dots here. Now, from top to bottom, again, the substrates show increasing sensitivity to CYP3A metabolization. And marked in yellow is celecoxib, which is essentially metabolized by CYP2C9. So here, phenytoin has hardly any effect, whereas rifampin, which is an inducer also of CYP2C9, does have a strong effect. So explaining the difference maybe between the two. And again, marked in blue at the bottom is the clinical substrate midazolam, which was also found to be similarly affected by phenytoin and rifampin, like we showed just for carbamazepine. And finally, in this table, we listed several substrates for which drug-drug interaction studies or data were published that were done with both carbamazepine and phenytoin, not as a direct head-to-head comparison, but at least giving a bit of an illustration of how comparable effects were. And this shows that there are both suitable alternatives, as it seems, having pretty strong effects on drug exposure. So there could be good alternatives, as it seems, for rifampin in drug-drug interaction studies. Now, this table is just to show you the experience that we have in recent years at Celerion using phenytoin, which is, in our view, the preferred candidate. In the marked area, you can see that the vast majority of studies has actually only been initiated after rifampin was banned by the FDA. And on the very right, you can see, in as far as data were available, that the reductions in exposure established by phenytoin were also pretty strong. So, in conclusion, until uncontaminated batches become available of rifampin, rifampin should be replaced in drug-drug interaction studies, with, in our view, phenytoin being the best or the most viable option for healthy volunteer drug-drug interaction studies, but carbamazepine also has been applied by others. Phenytoin and also carbamazepine have been shown to be effective as an inducer of CYP3A4. And lumocaftor may be the next generation inhibitor, but there's still some data to be collected, I think. then I said the recommended dose for phenytoin would be 300 milligram on a daily basis for at least 2 weeks, which is found to be generally safe and well tolerated also in healthy subjects. And for carbamazepine, this is also well tolerated in general, but it should at least follow a dose titration regimen. Now, with that, I want to conclude. And for those interested, the link on the slide below or below on the slide will take you to the mini review that Sabine also referred to and that we published in Clinical Pharmacology and Therapeutics earlier this year.
Well, thank you very much for that insightful presentation. I would like to invite our audience to continue to send their questions or comments right now using the questions window for this Q&A portion of the webinar. I'd also like to direct your attention towards the handouts module within the GoToWebinar control panel, where you'll find some additional documentation related to today's presentation to download for some further reading. Now, I've already received some questions, so we'll get ourselves started with those. The very first question that I have here, I believe would be best directed towards Arnaud, and it would like to know, how long and which dose did you use in your DDI study with Rifampin?
Thank you for that question. Well, Rifampin has been used across many studies in our clinic, but also outside our clinic by others. And the typical dose for Rifampin is 600 milligram once daily. And I believe that in our trials, we usually dose it for 2 to 3 weeks before studying the interaction with the study drug. I hope that answers your question.
Excellent. Thank you very much for that answer. The next question here would like to know, why do you dose Phenytoin 3 times a day and not just once like in patients?
Yeah, I can take that too. It's indeed in patients, we clinicians will treat patients with once daily treatments rather than 3 times a day as we recommend. But then patients are treated chronically with phenytoin, so that will help improve the adherence to a treatment, ensuring that phenytoin is reaching or will keep reaching full efficacy in those patients. Now, the risk for such a regimen in healthy volunteers is that there's a larger tendency towards higher peak levels, which are associated with the potential for adverse effects, bearing in mind that phenytoin has a relatively large or narrow therapeutic window. And maybe also interesting to mention in that context that late this summer there was a modeling paper in Clinical Pharmacology and Therapeutics from Chen and others, and they also looked at modeling phenytoin at different dosing regimens, and they also said that, for instance, a regimen like 100 milligram given across 3 doses a day will help keep the frequency of exceeding the therapeutic window below 10%, and thereby also dramatically reducing the risk of a study in healthy volunteers.
Very good. Thank you so much for that. The next question here would like to know, does the EMA use the same acceptable intake limit as the FDA?
Yes, I can actually take that question. So the EMA actually uses a different approach or a different value for the acceptable intake limit. The guidance actually outlines 2 approaches that could be used to determine this limit. And so the first is because nitrosamines are a class of or a cohort of concern, there's a class-specific limit set at 18 nanograms per day. And that's for any nitrosamines that wouldn't have any carcinogenic data associated with it. The other way to do it is to do a read-across approach. So using the nanograms per day of a structurally similar type of nitrosamine. And so the EMA actually uses the nanograms per day for NDEA, which is 26 nanograms per day. The FDA uses the read-across approach as well, but they apply the NDMA, which is 96 nanograms per day. So a little bit more conservative for the EU acceptable limits than, say, in the US.
Very interesting. Thank you for that. Our next question here asks if you have, either of you, any experience with metformin DDI study alternative where nitrosamine contamination has also been flagged by regulatory authorities.
Yes, I can take that question too. So while metformin has been found to contain some nitrosamines, it was actually only in certain batches. So there are batches that are available free of potential nitrosamines that can be applied for current DDI studies. So it's really not an issue when it comes to DDI studies or even just metformin dosing for patients in general. There are clean batches available.
Wonderful. Thank you so much. The next question I have here, can the less than lifetime exposure be used for nitrosamines to determine acceptable intake limit?
That's a great question, and if you don't mind, I'll take that one too. So the less than lifetime exposure is really for short-term or intermittent exposure, not for chronic continuous daily exposure. And in order to calculate this, a multiplying factor is actually applied to the nanograms per day for the determining an acceptable intake level. Now, because nitrosamines are this cohort of concern, the regulatory agencies actually don't favor this approach. Both the EMA and FDA have indicated that the less than lifetime limit would actually expose people to potentially higher exposure levels, even if during a shorter period of time, that would result in an unacceptable excess risk of cancer. So, like for example, with rifampin, if say dosing was less than 1 month, the NMP value using less than lifetime would be about 12, 13 parts per million, which is 2 times more than what the FDA has set as a temporary limit of 5 parts per million, and that's actually 80 times higher than what is currently mandated for healthy volunteers. So using this approach is not acceptable at the moment.
Very interesting. Thank you so much for that answer. The next question here first states that I remember that last year an FDA staff member recommended using carbamazepine and in a presentation. They want to know what is the FDA's latest position on what to use as an alternative of rifampin as a CYP3A inducer.
I can take that question. I think the FDA may, like many others, may look for alternatives, but I think in the communications we have with the FDA, they try to stay away from being very specific. And in fact, using phenytoin, which is the preferred candidate that we are using, has never been objected against by the FDA. So I think, like many others, people are searching for alternatives, and carbamazepine and phenytoin, like or like I showed in my slides, are pretty similar in being effective as an inducer, and under certain conditions can be safely applied in healthy volunteers. So I don't think the FDA has a preference for either. Sabina, please add if you have another update on that, but I don't think there's any communication officially that they prefer one above the other. So both are fairly exchangeable, and probably really depends on a site's preference. And as I said, we tend to use phenytoin instead, being not having a black box warning, for instance.
All right, very good. Thank you. The next question says that in one of the case studies, only the rifabutin part was done. Would this be an appropriate alternative using only a moderate inducer?
Sorry, was that right? Did you say rifabutin?
Yeah, I guess in one of the case studies it says here, so the
Okay, yeah,
rifampin part was done. Yeah.
yeah. So sorry, yeah. Um, well, yeah. In that particular case, rifabutin was planned next to rifampin, which is a strong inducer, and like the ICH recommendation that I alluded to earlier, is saying that you should actually use a strong inducer, but if you have no available, then you may switch to a moderate inducer instead. Now, with strong inducers available, we feel rifabutin is not a good alternative. But I think in this particular case, you could argue that and that's for any drug. I think that is uh, very probable to be strongly influenced by CYP3A inducers. Then I think studying rifampin may be a bit of an open door, and you may address that in your labeling anyway. So choosing for only studying a moderate or weak inducer may address your question in that case, but otherwise may yield inconclusive results because you still are not sure if you only have a moderate effect on your PK. So you may miss it with only a moderate inducer. So I would say no, it's not a good alternative. Yeah, so I hope that answers your question.
Very good, thank you. I think it does. Our next question here is curious that there are many other drugs in the market that contain NAs at different levels, and some of them do not yet have an established AI. Should we take this in consideration generally for any DDI study?
Great question. And what I will say is that there is the guidance that came out for the control of nitrosamines, and when there is no carcinogenic data, then the 18 nanogram per day cutoff could potentially be used, or as we talked about, the reach across method for identifying potential acceptable intake limits. But then I also do want to say that with this guidance and the requirements about identifying risk and then also following up on that risk, so determining the amount and levels of potential nitrosamines within medications and drug compounds, that so far this has brought about at least on the EMA, the data showed 60% that there was an increased risk. They've also showed that some of their manufacturers have actually responded and replied, indicating that only 2% have actually found nitrosamines to be above the acceptable intake level. So I think this is really the potential like known suspects like rifampin and rifampin. So in short, when it comes to DDI studies and using carbamazepine or phenytoin, for example, I think the regulatory agencies would have already signaled that there may be a potential risk there. And given that these compounds don't really contain secondary amines, I don't think that they will hold risk of containing impurities unless there are potential changes to the manufacturing process, which introduces these impurities. So in general, I believe that there is no risk there.
Wonderful. Thank you very much for that detailed answer. The next question here asks, how long will temporary limit for MNP be valid? So in other words, what they're wondering is by when can we expect to have an MNP-free rifampin available?
Yeah, that's the question I think we would all like answered. We know from the guidance that the step 2 and step 3 process should be done by October 2023. So hopefully by then we'll have some more information about the status of MNP in these batches and to see if they are below the temporary threshold or back to acceptable intake levels. I'm not too sure. Unfortunately, I can't answer how long this temporary threshold will be in place. I think we just kind of need to wait and see.
Very good, thank you. Next question here asks, so you showed that 16% of drugs pose a risk of nitrosamines. The question is, how concerned should we be with contaminated drugs reaching the market?
So that actually, I did speak to that question, and again, I don't think we should be too concerned. The guidance that is available has already included steps to address these, and I think all of the known potential suspects have been identified so far.
Excellent. Very good. Thank you so much for that answer and for all of the answers today. However, we have reached the end of the Q&A portion of the webinar. Now, if we couldn't attend to your questions, the team here at Celerion will follow up with you, or if you have further questions, you can direct them to the email addresses that are up on your screen. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from X-Talks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it will help us to improve our webinars. Now, I've also sent you a link in the chat box, and with this link, you'll be able to view the recording of this event on this page, and you can also share this link with your colleagues when they register for the recording here as well. So I encourage you to do that. Now, please join me once more in thanking our speakers for their wonderful time here today. We hope that you all found the webinar informative. Have a great day, everyone, and thank you for coming.
Thank you.
Thank you.